Welcome back to the deep dive. Our source material today is pretty dense, but it's structured in a way that's perfect for what we want to do. We're going to extract the absolute core mechanisms of clinical immunology. So we'll be drilling down on the molecular pathways, the diagnostics, the key immunodeficiencies, all the things that really matter in a clinical setting.
And that's the key, isn't it? It's so easy to get lost in, the alphabet soup of cytokines and cell types.
Absolutely.
What we want to do is translate those molecular events, whether it's one enzyme failing or the shape of an antibody, directly into what you see in a patient. If you understand the why, the clinical picture just follows.
Okay, let's unpack this then, starting at the most fundamental level, the blueprint of specificity.
The antigen receptor.
Exactly. Specifically, B-cell receptors or immunoglobulins. We know the basic structure is two identical heavy chains, the mu, delta, gamma, alpha, epsilon types, and two identical light chains, either kappa or lambda.
And right there, inside that structure, is a really critical distinction we have to make.
Idiotype versus isotype.
Yes. And I know these terms can get mixed up easily.
They really can, so how do we keep them straight?
Okay, think of it like this. The idiotype is the what. It's a lock and key. It's determined by the variable regions, and it defines what antigen the antibody can bind to. Specificity.
Got it. What it binds.
Then you have the isotype. The isotype is the how. It's determined by the constant domains of the heavy chains, and it dictates what the antibody does after it binds.
So does it activate complement? Does it get secreted into mucus? Does it cross the placenta? That kind of thing.
Exactly. It's effector function.
Yeah.
So you can have the same target, the same idiotype, but a totally different weapon, a different isotype.
That's a very clear way to put it. Now, what about T cells? The T cell receptor, or TCR, has a similar variable and constant structure, right? Usually alpha-beta chains.
It does, but its mode of action is fundamentally different.
How so?
Well, the difference is presentation. A B-cell receptor can just grab a soluble antigen floating by. A TCR can't do that.
Right.
Its variable domains can only bind to a little piece of a processed peptide that's being held out for it, presented to it by an antigen-presenting cell. It needs that context.
So we've covered how the system recognizes a threat. Let's shift to what happens right after an injury. The acute inflammatory response.
Exactly. You move from that specific recognition to just brute force. The body is damaged. The goal is to get the troops there fast.
Which gives you those classic cardinal signs: redness, swelling, heat, pain.
And loss of function. And to get the troops there, you need chemotaxis.
The chemical breadcrumb trail?
That's a good way to put it. The sources are great on this. There are 4 chemoattractants you just have to know.
Okay, what are they?
Leukotriene B4, interleukin 8, C5a from the complement cascade, and formal peptides.
And those peptides often come from the bacteria themselves, right?
Yes, so the invader is basically ringing the dinner bell for the phagocytes.
So once those phagocytes arrive and engulf the pathogen, the real war starts inside the cell. We're talking about the oxygen-dependent killing mechanisms.
It all kicks off with a respiratory burst.
Which is like flipping a power switch. This activates a membrane-bound enzyme, NADPH oxidase.
And that enzyme takes oxygen and reduces it to the superoxide anion, O2-. That's the first step.
From there, you get other toxic radicals like hydroxyl radicals and hydrogen peroxide, H2O2.
But the real heavy hitter, the ultimate weapon, is the cellular bleach.
Myeloperoxidase, or MPO.
Exactly. MPO comes from the phagocyte's lysosomes. It takes that hydrogen peroxide and combines it with chloride ions, just simple chloride, to produce hypochlorite, HOCl.
It's an incredibly powerful system.
It is. And while the phagocyte has other tools, oxygen-independent things like lysozyme and defensins.
This oxygen-dependent pathway is the most lethal, and we'll see later why knowing that first step with NADPH oxidase is so critical clinically.
Right. So moving on to humoral immunity, let's look at the trade-off between the early responder, IgM, and the more refined later one, IgG.
IgM is the brute force antibody, right? It's the first one made, and it's absolutely massive.
It's a pentamer. That means it has 10 identical binding sites.
A valence of 10.
Which gives it the highest avidity or total binding strength. It's also the best at activating complement. But there's a big functional sacrifice.
Because of that shape.
Yes, the pentameric shape physically blocks its Fc tails. They're sterically hindered. So IgM cannot act as an opsonant. It can't flag cells for phagocytosis.
So super strong, but not very versatile. Then you have IgG, the workhorse.
I'd call it the gold standard. It's an excellent opsonant. It supports ADCC, where it flags cells for NK cells to destroy. It activates complement.
And most importantly, it's the only one that can cross the placenta.
That's the one. It undergoes active transport across the
Yeah.
placenta, which is how a baby gets natural passive immunity from its mother.
So that switch from making IgM to making IgG is obviously crucial. How does a B cell refine its weapon like that without losing its target?
Two beautiful mechanisms. The first is isotype switching.
This is driven by cytokines, it's unidirectional, and you can't go back.
Correct. The B cell literally changes the constant region gene it's using. And you need to know the signals.
I think there's a mnemonic for this.
There is. Interleukin-5 causes a switch to IgA synthesis. Think 5 always. And interleukin-4 causes a switch to IgE synthesis. Think forever.
5 always, forever. Got it. So that changes the weapon. The second mechanism improves the binding itself, right? Affinity maturation.
Precisely. As the B cell is rapidly dividing, it intentionally introduces random mutations into its variable domains.
That's somatic hypermutation.
Yes. It's like rapid, localized evolution. The clones whose antibodies happen to get a better grip on the antigen are selected to survive. The ones that fit poorly just die off.
So you end up with an antibody that has a much, much better fit.
A much higher affinity.
All right, let's pivot to when this whole system goes wrong. Let's talk hypersensitivities. Gell-Coombs classification gives us the framework here.
Type BI is the one everyone knows. Immediate hypersensitivity. Yes. It's IgE mediated. The IgE coats mast cells and basophils, and when the antigen comes back, pollen, peanut protein, whatever, the cells degranulate and release all these potent mediators.
And that's what causes the symptoms. Histamine, prostaglandin D2, and the leukotrienes C4, D4, and E4.
Then you have type 2. This is cytotoxic or non-cytotoxic.
So here, IgG or IgM is binding directly to an antigen on a cell surface.
Right. And that can either lead to cell destruction, like in hemolytic disease of the newborn, or it can be non-destructive. The antibody just gets in the way of a receptor, like in Graves' disease.
Okay, type 3 is a bit different conceptually. Immune complex reactions.
It is a big shift. Here, you're not attacking a fixed cell. You have these circulating clumps of IgM or IgG and antigen that just get stuck.
But usually in small blood vessels, like in the kidneys or joints.
And wherever they deposit, they activate complement and cause this really nasty localized inflammation. Think post-streptococcal glomerulonephritis or serum sickness.
And finally, the outlier, type 4, delayed type hypersensitivity.
The outlier, because there are no antibodies involved at all. This is purely T-cell mediated.
And that's why it's delayed. It takes 48 to 72 hours.
Because you have to recruit and activate the specific T cells, Th1 cells, cytotoxic T cells, macrophages. This is the mechanism behind a TB skin test, poison ivy rash, and even granulomas in rheumatoid arthritis.
Okay, to really drive home the clinical impact of these failures, let's go back to that innate immunity-killing cascade. We have to talk about chronic granulomatous disease, CGD.
Absolutely. The molecular basis here is a deficiency in that first enzyme we mentioned, NADPH oxidase.
So the patient can't make the initial superoxide anion. The whole oxygen-dependent killing system just fails.
Which sounds catastrophic, and it is. But here's the clinical pearl you have to remember. These patients get recurrent infections specifically from catalase-positive organisms.
Okay, let's break that down. Why is catalase-positive the key?
Because catalase-positive microbes like Staph, Nocardia, Aspergillus, Pseudomonas, they have an enzyme, catalase, that breaks down hydrogen peroxide.
Their own hydrogen peroxide.
Exactly. In a normal person, a little bit of H2O2 from the bug could maybe still feed into that MPO system. But in a CGD patient, they can't make their own radicals, and the pathogen destroys its own peroxide. So both oxygen-dependent systems are completely dead in the water.
Wow. Okay. And the diagnostic test for this?
It's a functional test, the nitroblue tetrazolium dye reduction test, or MBT test.
How does it work?
A working phagocyte will reduce the dye, turning it from yellow to a purple-blue color. In CGD, with no NADPH oxidase activity, the dye just stays yellow. A negative result is the diagnostic one.
Now let's look at a parallel failure in humoral immunity, X-linked hyper-IgM syndrome. This goes right back to what we were saying about isotype switching.
It does. The molecular problem here is that the patient's T helper cells are missing a key protein on their surface called CD40 ligand, or CD40L.
And that's the go signal for B cells to isotype switch.
Without it, there's a functional block. The B cells can only make the default antibody, IgM. So they have tons of IgM, but dangerously low or absent IgG and IgA.
Which clinically means recurrent infections.
Especially from opportunistic bugs like Pneumocystis jirovecii pneumonia.
Okay, let's shift gears completely to the lab. How do we measure all these things? Let's start with the simplest techniques, precipitation and agglutination.
They both work on the same basic principle. You want antigen-antibody complexes to get so heavy they fall out of solution.
The only difference is the antigen.
Right. Precipitation uses a soluble antigen, like a protein. Agglutination uses a particulate one-think whole red blood cells or latex beads that you can see clumped together.
And for both of these, concentration is key. You need that Goldilocks spot, the equivalence zone.
Yes, the perfect ratio of antigen to antibody where you get maximal clumping or settling. Too much of either one and the complexes stay small and soluble.
Now, a cornerstone test, especially in the blood bank, is the Coombs test.
The antiglobulin test, it detects IgG antibodies on red blood cells.
And it's crucial to separate the two types. First, the direct Coombs test.
The direct Coombs answers the question, is there IgG already stuck to this patient's red blood cells right now?
So it's diagnostic for something active, like autoimmune hemolytic anemia.
Exactly. The indirect Coombs test, on the other hand, looks for free-floating antibodies in the patient's serum that could attack red blood cells.
Which is what you do for transfusion cross-matching or in pregnancy to check for maternal antibodies that might harm the fetus.
Precisely.
Let's talk about the classic two-step HIV diagnosis. It starts with the EIA screen.
The enzyme immunoassay, EIA, is a great screening test because it's highly sensitive. It won't miss many positives.
But the trade-off for sensitivity is specificity. It gives you a lot of false positives.
Which is why you can never, ever confirm a diagnosis with just an EIA.
For confirmation, you need the Western blot.
The protein immunoblot.
Yeah.
This test physically separates the key viral antigens GP41, GP120, P24 by size. To get a confirmed positive, you need to see at least 2 of those specific bands show up.
But there's a huge clinical challenge here. What if you're trying to diagnose an infant born to an HIV-positive mother?
You can't use EIA or Western blot because maternal IgG crosses the placenta. The baby will be full of mom's HIV antibodies, so they'll test positive even if they aren't infected themselves.
So you have to bypass the antibodies altogether.
You have to go looking for the virus itself. The only reliable diagnostic test in a neonate is PCR, the polymerase chain reaction, to find the proviral DNA inside the baby's own cells.
Okay, last topic. Transplantation compatibility. First step. Always.
ABO, blood typing, and cross-matching. No question. You have to do this to prevent hyperacute rejection.
Which is immediate, irreversible, and mediated by pre-existing antibodies.
Right. Beyond blood type, you look at MHC class II compatibility. The test for this is the mixed lymphocyte reaction, or MLR.
And this is basically measuring how angry the recipient's T cells get when they see the donor cells.
That's a great way to put it. You're measuring the T cell response by looking for DNA synthesis.
How do you do that?
You add a radiolabeled nucleotide to the culture.
Yeah.
If the T cells are dividing like crazy because they see a mismatch, they'll incorporate that radioactivity into their new DNA.
So a high radioactivity count is a bad sign.
It's a very bad sign.
Aye.
The counterintuitive part is that the best match, the best donor, is the one that gives you the lowest radioactivity. Less division means less recognition, which means a better outcome.
Wow. So that brings us to the end. I mean, we've gone from a single enzyme like NADPH oxidase all the way to specific bacterial risks, and from how antibodies are made to why a standard HIV test doesn't work in a newborn.
It really shows how understanding the mechanism is everything. You connect those molecular dots, and the clinical picture becomes clear.
It really does. All right, now for your clinical application challenge.
Okay, here's a question for you to think about. Imagine you're managing a patient who is just diagnosed with chronic granulomatous disease. They're going to need lifelong prophylactic antibiotics. Based on the mechanism we discussed, specifically that catalase problem, which common bacterial pathogen would you immediately target with that prophylactic regimen, and why?
A great question to ponder. Until next time, keep digging into the details. That's where you'll find the best clinical insights.